Hello students, welcome to Plants and Animals of Southern California. My name is Catherine Scafidi, and today I will be talking to you about my thesis research on the effects of an invasive alga, Sargassum horneri, on the trophic dynamics of temperate rocky reefs. We can start off with a producer like this grass that is consumed by an organism like this grasshopper, which is in turn consumed by a predator, such as this grasshopper mouse, and that is consumed by an apex predator. And each of these organisms are getting a positive interaction with the trophic level below them because they are getting food and resources, but they are also having negative impacts on the organisms in the trophic levels below them because they are directly consuming them. In a marine environment, for my research specifically, I am not going to be looking at an apex predator, but I will have a producer, consumer, and a predator. So the producer will be algae, consumers will be invertebrate epifauna that live among the algae, and the predator will be the reef fish that forage among those invertebrates. A lot of studies have looked at these interactions between trophic levels, but not too many have looked at the interactions or these effects of a producer that is invasive on a predator like reef fish. So that is my main interest for my research. Now, how do these invasive species get to become invasive? In the marine environment, it is due primarily to these shipping pathways that freighter ships use to go from coastline to coastline. And so this map shows the frequency of the pathways that are used around the world, and you can see in some spots it's completely whited out because those are frequently used throughout the year. And this map is a really good representation of just showing just how much of the coastline is used and is touched by these freighter ships. Therefore, we have organisms that are capable of crossing these once natural barriers, and that's how they can become invasive. So an example would be in our own home port, the Port of LA in Southern California, which is one of the largest ports of North America. And we get a lot of shipping traffic from the coast of Asia or off of Japan. And so these ships are able to uptake organisms that are in the water at one destination, such as Japan, and they can carry them over. They don't do it on purpose, but they get caught in the bottom of the ships, and they carry them over to, say, like our Port of LA, where they offload that water, and the species are able to survive in our waters if they can handle the conditions. And that's when we get these invasive species taking hold. So my study organism is Sargassum horneri, which was found in the Port of LA in 2003, and it's native to South Korea, Japan, and China, and it has an annual life cycle, which means it is capable of reproducing every year. It also has male and female organs on the same individual. So what that means is even if the individual is dislodged from the rocks, it can still float and drift and reproduce with itself. So that makes it a really good invasive because it can still propagate and spread. It also is a brown alga, therefore it contains these defensive toxins known as phenols. An example of phenols, it would on a terrestrial level would be like salicylic acid, which is found in willow trees. So these brown algae contain a variety of these phenols that can have various effects on different organisms. So my overall question for my research is, what effects does this invasive have on multiple trophic levels? And to start off, I have 3 hypotheses that I'm testing. The first one is whether or not the fish are foraging among it. So I expect them to have a lower foraging rate among the invasive compared to the native species, and that is due to a lower abundance of the invertebrate epifauna that live among the algae. And that is due to a higher concentration of these phenols that the invasive has, which is deterring those invertebrate epifauna from living among them. So my study sites are off the coast of Southern California on Santa Catalina Island, on the west end near the Wrigley Marine Science Center, And I have 6 sites spreading along that side, which is the leeward side of the island, which has had the longest invasion of Sargassum horneri since it was first found in 2006. For the first part of my study, I conducted fish foraging observations of rockgrass, California sheephead, and Garibaldi. And for this, I selected individuals and followed them around via scuba, and I recorded every bite they took and what alga that was associated with. So my results of the fish foraging are as follows. I have my algal species listed on the x-axis, and I have the foraging rate by number of bites per minute on the y-axis. And on the x-axis, I start off with the 3 native abundant brown algae, and then I have the invasive on the far right. And so overall, we can see here that there's this trend of the fishes foraging more among the native species and significantly less among the invasive species, especially they preferred to forage the most among the native Sargassum species, which is closely related to the invasive species, but they still were avoiding the invasive species. So for the second part of my study, I wanted to assess the invertebrate communities on these algal species, and to do so, I collected individuals of each alga, the invasive and the 3 native species listed below, and I collected them in these fine mesh tea bags where I could ensure that each individual alga I collected was also collecting the invertebrates that were directly living among and associating with that individual. I then took that back to the lab where I rinsed off the inverts and I was able to count and identify them under a microscope. I have my epifaunal abundance on my y-axis and again my algal species on my x-axis. There is just a significantly lower abundance of inverts on the invasive alga compared to the 3 natives. So this to me tells me that this is why the fish just weren't foraging as much among the invasive, because their prey just wasn't there. So then to test whether or not the phenols were responsible for the invert abundance differences, I extracted the phenols from the invasive and the 3 natives, and I used a Fullen method where I dried out the algae and then I was able to process the extraction. For this, my absorption is on my y-axis, and again I have my algal species on my x-axis. And so the absorption values, you'll see that the higher the absorption means that the greater the concentration of these phenolic compounds were in that algal species. So when we look at this graph, first off you can tell that the native Sargassum species has the lowest concentration of these phenols compared to the other 3. And so looking at just the native Sargassum versus the invasive Sargassum, there is a difference, and that could be why we saw the fish foraging among the native so much, because there are a lot of inverts living there, whereas the invasive had barely any inverts, the fish weren't associating with it. But there is this weird trend in the middle with the 2 other native species that they have equal, if not more, phenols in their tissue. And so what this could be is that they have these higher concentration of phenols that could be different types of phenols, and that the inverts living among those are adapted to those specific phenols and can live among those algae, whereas they can't transfer over to the invasive and live among that because the phenols the invasive has in high concentrations could be a different deterrent that's stronger and they're not able to tolerate. So overall, I found that my effects of the invasive on the fish foraging were that they foraged less among the invasive compared to the native algae, and that was due to a lower abundance overall of invertebrates living among the invasive compared to the native algae. And I did find that the invasive had higher phenolic concentrations compared to the native Sargassum species, but not compared to the other 2 species. And again, that could be some sort of evolutionary difference between the inverts tolerating the phenols the phenolic concentrations of those other two algae. And there's a fourth part of my study that I'm working through right now where I collected female rockgrass at all 6 of my sites. And this study was basically to see how the growth and their reproductive output could be affected by this invasive. So my sites have varying densities of this invasive alga, and I wanted to see if the reduced prey items and the presence of that invasive is actually affecting their growth by extracting their otoliths, which are these small inner ear bones that you can you can age them by similarly to how you would count rings on a tree. And I'd also take out their gonads, and I can count their eggs and see what their reproductive output would be for that year. So overall, we can see that sargassum can have more ecosystem effects than just the direct competition with native algae. So when the El Nino came through in 2015-2016, it wiped out the kelp forest, and Sargassum horneri took over, and everyone was very worried that this invasive was outcompeting the native for space. But there's also these other effects that it can have on these different trophic levels and organisms that are interacting with it that's still important to study. And studies of trophic interactions in general in various ecosystems is very important to understand, because we can then use that to recognize the susceptibility of different ecosystems to different invasive species before it happens. And invasive species research as a whole is important because we can use that to inform policy of ways to prevent future invasions or mitigate effects of these invasions, because they are very cost effective to get rid of, and usually once an invasive species establishes itself, it's impossible to get rid of. And that's all I have to say about that. Done.